Hello and welcome to this seminar about the crosstalk between bone and the vasculature in diabetes and CKD. This is an e-seminar that is jointly organized by the Diabesity Working Group, represented by Sebastjan is a professor of nephrology in Maribor, and myself, who is a professor of nephrology in Copenhagen. My name is Mace, and the CKD-MBD group, represented by Professor Carlo Alfieri from Milan. And also, I would like to introduce our speaker, who is Maria Mace, who is a senior registrar in nephrology and a postdoc at the Department of Nephrology at Rigshospitalet in Copenhagen. Welcome, Maria. I look forward to have this meeting with all of you, and I hope that you will be participating with exciting questions, and I hope the panel will be able to answer the questions. So please, Marie, please give it a try. Thank you.
Thank you for the nice presentation. Thank you for inviting me to present here today. I'll just share my screen. So the talk of today is crosstalk between bone and the vasculature in diabetes and CKD. I have nothing to disclosure. So I think in the daily clinical practice, we all recognize the heart valves and vascular complications to diabetes and CKD patients. And in addition, these patient groups also suffer from increased fracture risk. This has been known for CKD patients for several decades, but it's actually more recently there has been a focus on this complication to diabetes. So the health of the cardiovascular system and the skeletal system seem to be linked. Back in 1946, I'm not too sure if you can see the reference over here, Maroon published in Radiology, an association between low bone mineral density and the calcification of soft tissue. And that was actually the first time this association was reported. And this has been followed by several studies showing this association of decalcification of the bone and the presence of vascular calcification. And even though this association has been known for decades, the mechanistic link between these two pathological processes are still not well understood. And here I've just listed to the right some of the more recent groups and recent findings in speculating in this area. So vascular calcification is classically divided into the intima and the media calcification. In the atherosclerosis, we have activation of the endothelium, and circulating cells, monocytes, enter the intimal space and trigger a cascade of events with differentiation of cells, accumulation of lipid, fibrous elements, and followed by calcification. And this process triggers inflammatory pathways and narrow the lumen of the vessel, which compromise the blood supply to the affected tissue. The media calcification lays here in the muscle area of the artery, in the vascular smooth muscle cell, which differentiate into a bone-like secretory cell. So this vascular bone-like cells begins to secrete extracellular matrix in which the calcium and phosphate are able to precipitate in. This type of media calcification is characteristic for metabolic diseases such as CKD and diabetes. And this is quite a busy illustration, but I just think it nicely illustrates just a subfraction of all the different mechanisms and factors that can trigger this shift in the phenotype of a vascular smooth muscle cell. And if I was just going to mention a few of them, we have the advanced glycation and projects in diabetes, which trigger this cascade. And in CKD, is characteristic for the disturbances in the calcium and phosphate affecting these pathways here. But independent of initial triggers, when in the transdifferentiation of the vascular smooth muscle cell is initiated, this induction of the transcription factor RUNX2 and BMP2 that then starts altering the phenotype of the vascular smooth muscle cell into this bone-like secretory cell. And I've also highlighted some of the mineralization inhibitors over here. And that is because of some of them has been focused for some clinical studies. The matrix GLA protein is a protein expressed in the vessels and inhibits mineralization. If, for example, you have a knockout model of this protein, you develop severe vascular calcification. And this to be active, this protein is dependent on vitamin K. And so in the CKD cohorts, there has been some trials studying supplementation of vitamin K to our CKD and dialysis patients to see if it could have a beneficial effect on the vascular calcification. However, these trials have turned out negative. Another one I just want to emphasize is the FIT-twin-A, which is a protein produced by the liver, and that actually works like a circulating chelator of calcium and phosphate, because one have to remember that our concentration of calcium and phosphate in the circulation is actually saturated. So we need these mineralization proteins to inhibit that these minerals crystallize. And FIT-twin-A creates a compound with calcium and phosphate, creating the CPPs, the calcium protein particles. And that has also been a focus in the CKD population, which is characterized with a higher a more burden of the secondary, more toxic CPPs. And magnesium hinder this transition from the primary and the secondary CPPs. And there has been some two randomized trials focusing on magnesium supplementation to these CKD patients with conflicting results, published in Jason by Ian Bresendorf and Sakaguchi. But vascular calcification is very present in our CKD patient. Here, Ida Sørensen from our department showed in the Copenhagen CKD cohort actually a very high prevalence of coronary calcification and also the abdominal calcification. And the two figures very nicely illustrate that with the failing kidney function, you do have more severe calcification in our patients. And I think diabetes is actually seen like a severe disease of the vascular bed. And since the 1990s, there has been a reduction in the deaths from diabetes and a reduction of death from cardiovascular disease in type 1 diabetes, as well as in type 2 diabetes. However, the cardiovascular disease is still the leading cause of death in these two conditions. But going back to the skeleton again and looking at the fracture risks, now we go back to the CKD patients. Here we see the hip fracture incidence by 1,000 patients a year, published by Sharon Rowe and C. Jason. Interesting, at a very younger age, these patients have a significant higher fracture risk, which is uncommon at this age group. And again, we see that the stages of CKD also divided into the fracture risk, with higher fracture risk with the poorest kidney function. Here we can see the dialysis population being the ones with the most severe bone disease. And Dieter Hansen showed in a Danish cohort also that even though that the kidney patients receive a kidney transplantation, they still actually have a quite significant increased fracture risk, even though they have gained a kidney transplantation, which actually I think underline the importance of this complication to CKD. And bone disease and CKD is quite complex and not well understood, and it is being classified into the TMB classification, with T stands for bone turnover, bone mineralization, and bone volume. And here you see the nice bone here, and the bone biopsy, the bone marrow here. And the two end of the bone turnover spectrum is the osteitis fibrosa, where you see an increased bone resorption in the bone, creating these cyst formations, which is characteristic for high PTH activity, uncontrolled high PTH secretion and activity is seen in late stages of secondary hyperparathyroidism. And also, so one is not surprised that this bone is quite fragile. The bone marrow also have some bone marrow fibrosis. But at the other end of the spectrum, from that very high turnover condition, you have the adenomic bone disease. And on the bone, you have a quiescent bone surface with no really any bone resorption and bone formation going on. And this has been associated with oversuppression of PTH by active vitamin D treatment. But also, it cannot. There's also some of these conditions cannot be explained by active vitamin D treatment, and it's not known really what the molecular mechanism are causing this bone condition. And then you have the osteomalacia related to vitamin D insufficiency, and here you see on the bone surface the osteoid that's thickened. And there's more of it, and it's not being calcified properly. This type of bone condition is rarely seen in our CKD patients. So what is it that we do see? We don't do bone biopsies in our patients to actually characterize their bone condition. And there's also a few studies actually performing bone biopsies, and I've just listed them, a few of them. So in our dialysis population, the predominant bone condition is actually a low turnover condition. And then you have also a fraction having still a high turnover and a normal bone turnover. And in this cohort where they looked at CKD4 and CKD5 on dialysis, the percentages are slightly different. But in the daily practice, the clinicians are guided by PTH level as a marker, a bone marker of the bone turnover in our CKD patients. So, in recent years, there's been more focus on the increased fracture risk in diabetes. And I've just listed some of the relative risk factors that's been reported in the different meta-analysis. And just overall, I think it's a little bit clear that type 1 diabetes do have a more severe bone effect on the bone with a significant higher relative risk in comparison to type 2 diabetes, which however also have increased relative risk for factors. And the mechanisms causing this bone fragility in diabetes is not well understood. However, there are several factors in the two different conditions that will have a negative effect on bone. The type 1 diabetes is characterized by low bone mineral density, and insulin also have anabolic effect on the bone. So those are the two kind of thoughts around why this we have a low BMD, also because that type 1 diabetes usually comes at an earlier age before peak bone mass have developed. So these two factors in this condition may be one of the reasons why they have low bone mineral density. However, the level of the bone mineral density has been assessed not to completely explain the increased fracture risk in this condition. In contrast, type 2 diabetes, the patient usually have a normal or a higher bone mineral density. So that, at least out from a bone mineral density point of view, cannot explain the bone fragility in type 2 diabetes. And then I've just listed the differences between the two conditions that have that could have effect on bone. Some of it is not really it's not been really well examined, and you're not sure exactly what the mechanism are, but those are the ones that have been listed with the different proteins and hormones. But then they do share a similar things in their condition with hyperglycemia, advanced glycation end products, inflammations, cytokines, and the microangiopathy. And then the neuropathy and the fall risk is actually also come out in studies to have a quite significant factor on the increased fracture risk in diabetes. And the condition is actually characterized as a low bone tone of disease. And this is based on some marker studies and a few bone histomorphometry studies, but there's not that many looking into the really bone pathology. So we're going back to the bone and vascular access, because this is the focus for today's talk. There is, both in diabetes and in CKD, several factors with adverse effect on the bone and the vessel. And here I've just listed some of the few of them in the middle, with the endothelial dysfunction, alteration in hormones, the inflammation, the oxidized lipids, oxidized stress. And again, these characteristic for the diabetic patients, and more characteristic for the CKD patients are the alteration in the mineral balance, the increase in the toxic CPPs and uremic toxins. And at least in the CKD population, the bone vasculature access have mainly been explained that well, it's been seen like that it was the disturbance in the bone, that it was the bone disturbances that primarily triggered and exacerbated the vascular calcification you see in our CKD patients. And this is also supported with some experimental studies. If you remember the slide of the vascular smooth muscle cell, phosphate in itself is able to trigger this shift in phenotype and triggering this pro-calcifying environment. However, in a previous study from our group, we looked into the vascular calcification, and here we used we examined the calcified aorta here from a 5/6 nephrectomized rat, and examined them using RNA sequencing technique. And actually, approximately 1/5 of all genes are actually differentially expressed in this diseased vessel, illustrating what a significant impact this disease process has on the tissue. And we found activation of the Wnt pathway, and that has also been shown by several others. Wnt pathway is usually active during embryogenesis, and then this pathway is silenced in most organs. However, it continues to be active and an important anabolic pathway in bone. However, in vascular calcification and also in other disease processes in other organs, this Wnt pathway, this powerful pathway, seems to be induced and stimulated to be expressed again. And we also found upregulation of Wnt inhibitors and upregulation of TGF-beta ligands. And since that these Wnt inhibitors and TGF-beta ligands are circulating factors, And they also found an increased plasma levels in our CKD patients. We speculated whether the calcified vasculature would actually secrete these signal molecules, which then could have endocrine effect on the bone metabolism. So we developed this novel model of aorta transplantation, where the calcified aorta from a CKD rat was transplanted with an end-to-end anastomosis into a healthy rat. So if the healthy rat would have like an extra bit of the aorta. And then, of course, we performed an operation where we transplanted normal aortas. And this transplantation was done in isogenic rats, so we didn't use any sort of any form of immunosuppressive medication. And then 4 weeks after the transplantation, we examined the bone of the recipients. And the rats that were transplanted with a calcified aorta, which approximately will have like 2 centimeters of calcified aorta in its abdomen, had lower trabecular bone mineral density, had lower bone formation, had disturbed Wnt signaling, and action upregulation of the mineralization inhibitors. And I'm just going to show you some of the results. So I'll just show you some of the results from this study. So here you see the bone mineral density of a normal rat age match. And the rats that received the normal aorta had this bone mineral density. So there was an effect of the transplantation by itself. However, the normal rats that received a calcified aorta had significant lower trabecular mineral density after 4 weeks. This was the area examined. And at this point, we could not detect any differences in the trabecular number and spacing. But the lower bone mineral density was supported by significant upregulation of mineralization inhibitors in the bone. Here we see osteopontin, which was significantly upregulated in the rats that had the calcified aorta, and also ACT, which regulates extra cellular pyrophosphate levels, which is also a mineralization inhibitor, was also significantly upregulated. And then we in the genetic analysis by using PCR, we found several changes in bone formations, genes related to bone resorption and related to bone cell communication. And I just want to just show you one of these results is actually the sclerostin, because sclerostin is the bone's, the local bone inhibitor, Wnt inhibitor in bone, having a negative negative impact on the Wnt pathway in bone. And that was significantly upregulated here. And to translate, because I quite like to translate these results into a nice bone histoformatory bone biopsy study done by Grazioli, where then in their bone biopsy study found that actually at CKD2 and CKD3 patients had a significant upregulation of the sclerostin in their bone, and also a significant increase in phosphorylated beta-catenin, which is a sign of inhibition of the canonical Wnt pathway. So at this stage, there seems to be an upregulation of sclerostin. And if we remember Ida Sorensen's Copenhagen cohort, quite a few of these patients at stage 2 and 3 actually had the presence of vascular calcification. So, but to further demonstrate a direct link between the vessel and the bone, we developed some in vitro studies where the aorta was cut into small rings and incubated ex vivo to detect secretion of signal molecules, and also develop a co-culture system where the aorta rings were incubated with a bone cell line. So first with the secretion model, we found strikingly high secretion of sclerostin into the media. And these are over 10 times the concentration that you would find in plasma in this rat. And also another Wnt inhibitor, the DCOF1, was induced and secreted by the calcified aorta. And also the TGF-beta ligand activin A had really high secretion by the calcified aorta. And then I've just put in some pilot data because we looked a bit into like the inflammatory pathways and measured interleukin-6, which also had really high secretion of this signal molecules. And all these signal molecules here have a negative regulators of the bone metabolism that could be potential some of the signal from the calcified vessel to the bone. And then in our co-culture system, where we incubate the bone cell, like a bone cell-like cell line, we use the UMR 106. And here you see the bone cell line, the mineralization developing. When the bone cells were incubated with the normal aorta, we did find significant lower mineralization numbers. Well, numbers of these, we calculated and measured the size and the numbers of all these mineralization units. But strikingly, when the bone cells were incubated with the calcified aorta, we could not detect any calcification at all. And this actually showed the quite powerful effect, anti-mineralization effect, that these secreted signals from the calcified vasculature has on the bone mineralization. So to sum up, our studies do suggest that the calcified vasculature do play an active role in the vascular bone disease, with factors secreted from the vasculature that via the circulation reach the bone, cause lower bone formation, disturb mineralization, upregulation of mineralization inhibitors, and altered Wnt and other signal pathways in bone. And then in turn, the disturbed bone metabolism will then probably, most likely, exacerbate the vascular calcification. So you have this negative crosstalk in CKD, like a negative spiral of demineralization of the bone, mineralization of the vessels, and so on. So I think I've used my time now. And these are just the acknowledgements for my current group and the people who has been involved in these studies. But I think we probably have to move on to the question and discussion now. Thank you.
Thank you very much, Maria, for this excellent presentation. And the presentation, I am sure, is now open to a lot of questions. And I would like to begin to ask if Carlo and Sebastjan has some comments at this early stage.
Yes, absolutely. Can I start?
Yes,
it's really compliments for the presentation. Really full of novelties and of good point. I have just consider that we are merging a Diabesity and CKD MBD working group. I have a question for you. I don't know if there is an answer, but it could be. That we saw that basically the inflammation and the inflammatory processes are really important in these processes. Do you think that the novel diabetic drugs, I mean the inhibitors of the glucose transport, sodium glucose transporter, but also the GLP-1 that maybe have more powerful in the reduction and modulation of inflammation could have a future place in these pathways, in these processes, in the modulation, obviously.
I'm not too sure if I can give you a good answer to that. I think it's a very good suggestion. I think that the organ protective mechanism of the SGLT1 inhibitors, I think not only been explained by alteration in hemodialysis. I also think there's something in the cell metabolism that's being altered there that could also probably be beneficial in other conditions. But I think maybe it could, but of course you need to do the studies for it, and so it's always difficult, if you have mapped the molecular mechanism in a disease process, it's easier, to go in a specific target. So there is two approaches to it. Instead of testing, you can test whether it works or not.
Sure.
SGLT2 inhibitors, I think, was a little bit of a surprise.
Yeah, my question was related by. My question was related by the fact that if you consider one of the first slides that you shared with us, there are a lot of pathways in which we know that SGLT2 have a modulation role, and also the GLP-1. So it could be really interesting to evaluate, because effectively it could be a point of connection between the bone and the inflammation also.
Yeah.
As far as I know, there was a study published recently about canaflozin, showed directly what you already said before, Carlo, so that this downregulation of some signaling pathways is already available. So some results indicating that they're doing contra-inducing the
calcification.
Sorry, yes. So we have some studies at the moment available for canagliflozin, as far as I know.
Thank you very much for these preliminary questions. I also have a question to you, Maria. Can you translate these findings in animal models to human pathology to the CKD patient? Is it that easy to translate animal studies into human studies, or what is your suggestion to overcome this challenge?
It's not easy, because as we all know, a lot of the finding in the animal models, when we test it in humans, it doesn't have the same findings or the same effect. I do think that when you look at some of the human studies, that there is an indicator of that there could be a similar crosstalk going on in the human pathology. Some of these markers I've shown has been demonstrated in some human biopsy studies of the vasculature, that they are expressed in the tissue, but of course we don't know if that is being secreted. But we are just planning to conduct further studies and starting our PhD project now in our clinic to try to look into some of using human tissue ex vivo and examine that to see if we can do a similar setup and demonstrate the same things, because it is we do know that these processes are interrelated. Aging, diabetes, CKD, that's been shown for decades, and there must be mechanistic links cross-talking between those two organs. And I think what it has mainly been a focus on the disturbed bone metabolism, where you then have the excess calcium and phosphate from the bone because of bone resorption, and then that is being is calcifying the vessels, but I do think that we need to look a bit more into the vasculature and see if that plays a more active role, because we know the vasculature plays a very active role in all the complications to diabetes, for example, in the different angiopathy that they have in the different vascular beds. And so I think that's what makes this study interesting, in looking a bit more. And what I also think could be interesting of it is that it might be able to detect some of the early mechanisms, because I think that if you want to hinder the progression of it, you need to start a bit more earlier in the development of vascular calcification and in the bone disease in CKD.
Yeah, that is interesting you say that, because what is the right time to begin the prevention of the mineral bone disorder in especially patients with diabetes? When do you think that we should actually start screening? Are we too late for the present time to screen?
Well, I think when it comes to the CKD-MBD, I think, we know that the phosphate binders decrease plasma phosphate, but doesn't really have a significant effect on the cardiovascular outcome. We do suppress PTH secretion. Okay, there's limitation because there's a variance between patients. We don't know what the optimal PTH level is at that particular patient, if we are at a right turnover or if we are over suppressing PTH. And that has also, but keeping the bone resorption at a lower point does decrease the risk of fractures, but the patients still have an increased fracture risk. And I think when you look at the time scale of all the hormonal changes at the cause of CKD, with an early downregulation of growth or upregulation of FGF23, followed then by downregulated vitamin D, we actually attack this complication quite late in our CKD patients. And I think when the calcification has developed, it's not reversible. We have shown that with when we looked at the aorta that was transplanted, some of the fibrosis and things that normalized, but the actually calcification that persists in that vasculature. And that is also in transplantation studies of CKD patients receiving a kidney transplantation. They might not progress as fast as with their vascular calcification, but the established calcification is not reversed. So I think that we need to look at some of the initial factors to try to hinder it to be established, but then also, of course, find something that can, like a pan inhibitor of its progression. And then again, that is complicated because, as I demonstrated, the calcification of the vessel is very similar to calcification of bone. So if you have a non-specific treatment, then you will most likely affect the bone as well. I think we can see that in the new drug for calciphylaxis with the SNF 476, I think it's called. When you look at those primary data, it hinders the calcification, but when you look at the bone data, it is also affected by it. So I think that, but we're not able to say when to start now, because I don't think we have actually identified when this complication to CKD is problematic and when it's starting. I don't think that we have actually really identified that point. And I think you need to first identify that point to know when you're treating it at the right time. And when it comes to diabetes, I think there's not, you do say that, if they're classified as an osteoporotic patient, you should consider giving anti-resorptive drugs. And there has been some in the osteoporosis trials, there has been some diabetic, but it's mostly postmenopausal women, and they do gain the same increase in bone mineral density. But again, I think you need to look a little bit more into what is actually the bone complication to this condition, because when you look at it, it doesn't seem to be an uncoupling of the resorption and the bone formation. It seems more to be a problem with the bone formation. So I think, again, it's difficult to address this complication now in our clinical setting.
Yeah, I think you really exclusively answered my question, I must say. I think Carlo has a comment also.
Yes, I have I have a comment about that, but it's just my opinion. So if we consider the best uremic toxins that we know today, we know that is phosphorus. So in my opinion, the treatment of the CKD-MBD, that means vascular calcification, PTH, and bone disorder, should start from the beginning of the CKD. Exactly when the protective mechanism that we identified in FGF23 and PTH starts to increase. Okay, what I mean is that the simply prevention for our patient that should be started from the beginning could be just to regulate the nutrition, to educate them to avoid the high phosphate food. Okay, but just from the beginning, from the point in which the mechanism that are protective start to increase, and this is what we usually do, and I think what is correct to say generally.
Thank you, Carlo. Carlo, I think there is a question from the audience saying, how do you differentiate internal from medial calcification in the clinical setting? Is that possible?
Well, I haven't.
So,
you go.
Is it for me? I didn't understand. Sorry.
I think it's for everyone, because there is a, it's a very general question from Ahmed El Husseini.
Yeah, of course, we have different tools to find out where the calcification is. So we have non-invasive, invasive tools to search for, but of course they are cost and not so easy to reach everywhere. So the answer will be, it's not so easy in the daily clinical practice, but we can speculate according to some pulse wave velocity, according to some intermedia, according to some calcification scores with CTs, where we are and what kind of problem we have. And of course, if we have increased bone disease also weight, so we can speculate that this is also the problem with the media. But directly to answer this is not so easy in the daily clinical practice.
No. And there's also another question about from Jordi Bauer, who says, is there any direct relationship between inflammation and sclerosing? Is there between activin A and inflammation? Are they independent? I think this is directed to you, Maria.
So I'm not too sure if I understand the question. If inflammation and activin A are interrelated?
If it is the relationship between activin A and inflammation and independent.
Yeah, I think when you have an inflammatory process going on in the tissue, activin A is induced.
Okay.
And in injured tissue, activin A is induced. And yeah.
Okay. And there's also another question from an anonymous attendee, and it's about the vascular calcification. Are there any studies related to wave shock for treatment of this condition? I think I don't have any idea of wave shock treatment for this disease. I think
Not to my knowledge.
Studies about this? No.
I heard about the terms, means that you have some device that you can have the waves who can do the mumbo jumbo with this calcification inside the tissue. But I'm not sure, but I read somewhere about this kind of treatment.
And then there is a question about the early markers of the crosstalk, Maria. Can you differentiate between the transition of CKD from stage 3 to stage 4? Are there any markers that differentiate between the different stages that you would bet your money on?
At the moment, I'm not going to bet any money, but we are actually looking a bit into doing a longitudinal model where we try to see assess the secretion from the vasculature to the time of being in a uremic state. I think from a theoretical point of view, I would think that the markers probably differ if you are at the first stage of the initial transition of the vascular smooth muscle cell into a bone-like secretory cell. I think that this cell will secrete different markers. When it is a bone-like cell, I think it would probably secrete more markers that are found in bone, where maybe at the earlier stage you would have more of those signal molecules. But that is all speculative. But hopefully we will find it out. And it's difficult to do human studies like that, but we'll start with an animal model looking into that, and then hopefully we'll find a way to translate these results into to human pathology.
Okay. Then there is a question that how should we use the prophylactic measures? What is the so to see the right ways to stop osteoporosis in CKD? So what is the right sort of ways to start it? What is the right recipe to go? I think this is a clinical question from the audience. Carlo, will you start?
Yeah. Okay. Yeah. Probably. So the first point is to make a photo of the mineral metabolism status of our patient. So if we consider, for example, the CKD stage 3, 3A or 3B, obviously we have to consider all together, and not just one time, but in the timeline of our patient, the most important, basically, mineral metabolism parameters that are the vitamin D status, the PTH. I think, but I ask also to all of you, what is your opinion, the bone turnover markers? And the first thing to do certainly is to solve the hypovitaminosis D. Usually we use supplements of native vitamin D. Be careful, obviously, to calcium and phosphate levels. And then and this is the first point, the first real point. Obviously, in patient with high risk for fracture, I think patient with the previous steroid therapy or with a strong osteoporosis, the use of bisphosphonate is absolutely mandatory. And when it's not possible to use them, denosumab could be useful. But it's really difficult, in my opinion, to give a guidelines when the actual guidelines are really not specific, because I think that each patient has specific characteristics. So, but usually we can generalize in this way. But I'm curious to know the use of the bone turnover markers in these settings. What is your opinion?
Are you asking me about bone turnover markers?
No, I ask to everybody.
To everybody, yeah. But I think it will be helpful in a clinical setting having more marker than just PTH. I think Henna Jørgensen and Peter Effinable have published some very nice studies on the use of other markers, and I think that can probably guide one's evaluation of the individual patient. And I also think it's difficult to give the right prescription on how to deal with this complication, because I don't believe that we today have the ideal treatment of this complication, because there's still such a big gray zone of unawareness of what's going on. But I think if you improve the bone health, you have a better buffer capacity. And in that setting, you can probably buffer your excess phosphate better, which I think will be beneficial for the patients. But I also do think, and that is very speculative, that we've been focusing on PTH and phosphate for many years by now. And if that was only that we need to focus on, I think you would have expected better outcomes. I think we need to look a bit more into other pathways. For example, the Wnt pathway, as I suggested here in this talk, and also maybe activin A and other pathways in bone, because we also see that in the bone biopsy study I showed. I know it's small group of patients, but it does illustrate that you actually at an earlier stage of CKD have quite striking alteration in your bone, and that is actually at an earlier stage where you don't have such a severe disturbances in the PTH level, in FGF23, suggesting that there are other mechanisms and other important factors to focus on in our CKD patients if you want to improve the bone health.
Carlo, you have a comment?
I totally agree with you. So stop to think only to the PTH levels.
Yeah, I want also to add, I totally agree also, that we have to think about measuring 25D level also, not to forget. This level, maybe the cutoff is a little bit too low, maybe, we will see. But again, I totally agree, PTH is not enough. Maybe it's enough to follow, to see the degree increase and how does it rise, but otherwise, not really. I will use now the opportunity, since I can talk, to mention the ideas to treat at least animals with a vitamin K or magnesium. Maybe Maria, do you have any experience, since you are expert on these animal models, for this kind of opportunities? Vitamin K2 or something like that?
Actually, I know other groups have used magnesium and vitamin K in animal models. I have not performed any of those studies myself, so I'm not the best to ask. But
sorry, okay.
So I have a question regarding the bone biopsy, because would it be that you actually need to early in a patient's life, maybe at the stage where the kidney function is closing to dialysis, or after being transplanted, that you should consider to make a bone biopsy. So what is the right time to consider this? Have you any thoughts about this, Maria?
I think I think that you can actually argue for a bone biopsy at many stages of CKD, but I don't think that patients will agree on that. I think that at the CKD stages, I think we need to do some clinical studies where we then look at our CKD patient at an earlier stage with bone biopsies, with also imaging tools to characterize what actually is their bone condition at CKD stage 3A, 3B, CKD 4. Because I don't think in a clinical setting now, it won't have any consequence, therapeutic consequence, unless you want to start up in an anti-resorptive medication and want to make sure you don't have an adenonic condition. Yeah, then you can argue in a clinical setting to take a bone biopsy. But otherwise, I think we need to do more research to understand this. These bone complication in CKD. And maybe could be able to correlate it to some tools that are a bit more accessible in the daily clinical practice. I think at kidney transplantation, it could be interesting to do a bone biopsy to see what type of bone condition they do have at transplantation, in order to maybe evaluate after transplantation whether they should be candidates for anti-resorptive medication. I think we have had a few cases where they actually had quite osteomalacia, which was found out quite late after they had fractured and had a bone biopsy. And so I think that could also be an indication here. But otherwise, it's difficult to say when it's indicated in a clinical setting. There's not that many centers who perform bone biopsy or able to get the biopsy evaluated. There's only a few centers that are able to do that.
Okay, thank you, Maria. So, Sebastjan and Carlo,
Yeah.
you have a question or a comment, maybe?
Just a small comment. The last question, maybe sometimes this bone alkaline phosphate level can be also useful if you don't know where you are regarding the problems in the bone. So if you are seeking also some osteoporosis problem behind. So sometimes to stick with the old markers also.
Okay. For now, I think if Carlo or Sebastjan or Maria, if you are finished with your comments and questions, then it's my pleasure to wrap up this very exciting presentation by Maria Mace that was looking into the crosstalk between bone and bone mineral disease and diabetes. We saw exciting news about the vascular-derived secreting factors, about lower bone formation, about disturbed mineralization, about upregulating mineralization inhibitors, and the signaling pattern that were altered. And we saw interesting results from the Copenhagen CKD cohort, from Maria's own studies of the topic. And for this, I thank you very much, Maria, for the presentation. And I hope that if you have further questions, you are welcome to write to the group, and hopefully this will lead to even further research questions. So I will ask you all if you have a final comment. Otherwise, it's my pleasure to close the session.